session on recombinant DNA technology in which we are talking about the genomic library construction or the cDNA construction. Now, complementary DNA is required if we want to understand the functional or expression part of the genome in a cell. If we want to know what genes are expressed, then we go for cloning the complementary DNA. Complementary DNA is produced by the reverse transcriptase of the mRNA which is formed from the different genes. So, looking at the cDNA cloning, the complementary DNA cloning for the preparation of cDNA library, we have the first strand synthesis is shown here. So, if you have got an mRNA, so in mRNA you know there is a poly A tail which is there. So, for the construction of mRNA, you use the primer sequences. The primer sequences which contain, for example, the T in this case, a long chain of T which will combine, which will form hydrogen bonds with the poly A tail, and you use the reverse transcriptase enzyme that will synthesize the complementary strand of the mRNA. So, this is basically the first strand synthesis that is required. So, this after this, so this is a DNA RNA hybrid. Now, this is then RNase H1 is used to digest the RNA. So, this is the first strand synthesis. So, initially a DNA RNA template is formed, then we use an RNase to digest the RNA and you get to have the single fragments which are there. So, you can see the single fragments which are there. For the second strand synthesis, you need DNA polymerase enzyme. Now, this DNA polymerase 1 will synthesize. So, all you can also see is when the RNase H is degrading the RNA, then it is leaving the small fragments of RNA still bound to the DNA. So, these small fragments then act as the RNA primers. So, you know DNA polymerase 1 will then synthesize the complementary DNA of the DNA template, that is the first strand that has been synthesized, using these small RNA fragments that will act as the primers. And after that, you will see that you have got a double stranded complementary DNA. So, this is termed as the cDNA because it is actually the coming from the mRNA. So, even if there were like exons and introns in the gene sequence, because only exon will come into mRNA, this is only the expressed part. And then you can go ahead, cut it with different and follow the simple recombinant DNA technology procedure. You can cut it with different enzymes, you can use a vector, and after that you can just clone it into a vector which is there. So, this will be forming a cDNA clone. So, for forming a cDNA library, you prepare cDNA clones like this. So, you isolate the total mRNA of the cell. From the mRNA, you construct a complementary DNA. For that, first you synthesize a DNA RNA template, then RNase H is used to degrade the RNA, and then the DNA polymerase 1 synthesizes using the small fragments of RNA as primers the complementary strand, and you get a double stranded DNA that can then be cloned into a vector. So, this is a simple procedure for screening for construction of the cDNA library. So, we looked at the construction of the genomic DNA library as well as the construction of the cDNA library. Now, when we have constructed the cDNA or when we have constructed the genomic DNA, we will come back to the point where we need or we want to sequence the entire set of the genome. Nowadays, we do not use such kind of a shotgun sequencing like we started with the Human Genome Project, but that is one aspect of it which can be done. But sometimes what we do is we also construct the entire library so as to identify a clone that is carrying our gene of interest. So, you carry the whole set of fragments, but that set of fragments, one of the fragment is your gene of interest. So, you have to screen the entire library for the desired clone. So, this is what is done, and there are different methods or procedures by which the screening of the desired clone can be done. The most common method is what is termed as the hybridization probing. So, by using hybridization probing from the entire set of recombinant clones that is representing a genome, we can representing a genome or representing an expressed part of the genome in case of cDNA, we can use this method to identify the clone which is carrying our gene of interest or our region of interest or the desired clone as it is termed as. Now, what do you mean by hybridization? to complement any nucleic acid strands hybridize to each other. This is a very basic property of DNA molecule. You know, the DNA two strands are complementary to each other. They run in anti-parallel direction. We know that. We have been studying that. We know that. We did it in molecular biology as well, that the typical structure of a DNA is a double helical structure where the two strands are complementary to each other and the two strands are held together by hydrogen bonds. So, you can see that when the two strands are held together by the hydrogen bonds which are there. Now, when we talk, and this holding together is termed as the hybridization. So, there can be some unstable hybrids, there can be some stable hybrids, and there is a DNA-RNA hybrid. What does this picture represent? This represents that if there is lesser complementary regions in the two strands, you will still have sporadic regions which are complementary to each other coming together, but that would basically be an unstable hybrid because there are many portions which are not complementary. However, there are only few portions which are complementary to each other. Then in the stable hybrids, if the two DNA fragments are highly complementary to each other, still they are not perfect, but they are highly complementary. You can see that still there are certain gaps which are there in between, but the two strands are very much complementary and they are forming the hydrogen bonds. So, such hybrids are termed as very stable hybrids. However, if you look at a DNA and RNA hybrid, so RNA is a complete complement to the DNA template strand from which it is being formed. So, if you look at the DNA RNA hybrid, it is a perfect zipper. So, the hydrogen bonds or the complementarity or the hybridization is very stable, and you know it is completely hybridizable, that is formed, which is shown here. So, this is the concept of hybridization when we talk about. Now, how to use this for the screening of a library? Now, when we are forming a library, depending upon the vectors we have used, either we will have the recombinant clones in the colony, in the bacteria. For example, the host organism is E. coli. So, if we introduce the recombinants in E. coli, what we will have is on the plate the different types of colonies which are there. You know, so these colonies, the entire set of bacterial colonies represent or contain the recombinant clones representing the entire library. But if we are using the phage vectors where the final recombinant clone is introduced into a phage that infects a bacterial lawn, then you know that it will lyse the bacterial lawn and create what are called as plaques. So, after the introduction of the recombinant clones into specific host, either you can have the colonies on the plate or you can have the plaques. So, if it is virus that is infecting a bacterial lawn, you will have the plaques, which are basically the zone of clearance on a bacterial lawn, or if you have the recombinant clones introduced into a bacterial host, you will have colonies. So, these two will be the outcome. Now, this is our library, either the colonies or the plaques, and now we need to identify that out of these colonies or out of these plaques, which is the clone that is carrying our gene of interest. So, this schematic representation again recalls how the plaques are formed. So, this is one scheme of cloning that I am just elaborating, that you isolate the entire genome or the cDNA, then you cut it into shorter pieces, you take the vector DNA, which is the phage DNA, you introduce them, or for example cosmids, although cosmids are introduced into E. coli as a host organism, but you take the phage DNA, you combine them together, you create recombinant molecules, you do the in vitro packaging, you package them into phage heads and tails, then you use them to infect the bacteria. When it infects the bacteria, it undergoes the lytic life cycle, and as you can see, undergoing the lytic life cycle, it will just degrade the bacterial cells. It will basically kill the bacterial cells and the viruses would be released. So, you will have the clear zone which is there. Now, each zone will have the recombinant plaques that would be, it would be a mixture of plaques, it will be a mixture of bacteriophages carrying the recombinant clones, but you will have the plaques. So, mixture in the sense on the plate. because we assume that when we are plating, then each plaque is coming from a single bacteriophage. So we count them as one only. So we had talked about this, so they are termed as the plaque forming units. So either the same way is the colony, the only thing is the introduction is inside a bacterial host, and when you plate it, you get the colonies. So this is the the outcome. This is basically the thing which you have as a library which you want to screen. So we will look at the mechanism of colony and plaque hybridization, that how to do it. So how the screening is done. Now again, as shown in the schematic diagram, the first step is to create the replica of this library. Now just remember that this library, as I said in the previous slide, it can be a colony or it can be a plaque. So on the plate you can have different colonies containing the bacterial cells containing the recombinant clone, or you can have plaques containing the bacteriophages containing the recombinant clones. So these are the recombinant DNA. So what you do is you make a replica of this plate onto a nitrocellulose membrane. So you can see that we can look at the procedural. This is a schematic representation. So you cut a nitrocellulose membrane and you press it slightly onto the plate, and therefore on the plate you will have either the cells, the bacterial cells, or you will have the bacteriophages that will stick to it. So this is basically shown for colony hybridization, but the same scheme is followed for the plaque hybridization screening also. So you can see what it does is there will be a replica which is on the nitrocellulose membrane. Then these cells which are now stuck onto the nitrocellulose membranes are then digested. So the proteases are used, alkali is used, it is digested on the membrane only, and therefore the DNA will be released there only at that specific position. Now this DNA will then be cross-linked, because now when we are adding the when we are doing the hybridization, when we are adding the complementary DNA, then it basically have to be stuck onto the nitrocellulose membrane. So for that stucking, different methods are used. For example, 80 degrees Celsius पे heat करते हैं, we generally do heating at 80 degrees Celsius, and therefore it will just cross-link the DNA onto the nitrocellulose membrane. So the DNA is stuck onto that, and then the cross-linking will also occur. So you will undergo these procedures so as to make DNA stably bound onto the nitrocellulose membrane. Now one important point to notice in this starting steps of the colony hybridization is that on the nitrocellulose membrane, it is the replica of the plate which is there. So the replica of the plate, so the positioning of the cells is same as that was found on the plate, because later on then you can go back and take out from the plate where the recombinant or which one is the recombinant clone, that you can find out. So this is basically the first part of it. The second part is then you introduce a probe which is a labeled DNA. Now what is probe? Probe in English means basically something to investigate. So this probe is actually investigating that out of the colonies or out of the DNA which is now on the nitrocellulose membrane, which is the recombinant DNA carrying our gene of interest. So this probe will be added and this probe will be labeled. So because this probe is going to hybridize, so hybridization require complementarity. So this probe will be synthesized from that gene of interest that we want to identify. Now depending upon, so sometimes it happens is that I have identified and sequenced one gene from one bacteria. Now if I am studying the same pathway in any other species, any another genus, then I might look at the gene which is present in my bacteria, is it present in that also, because they are also carrying out the same catabolic pathway. So for that what I do is I sequence the gene which is carrying out a particular pathway or a reaction, I sequence it and then use that sequence to create a probe and screen the genomic library or cDNA library of the other organism and see if it binds. Now there is a possibility that it will not contain the 100% identical gene of what my bacteria has. But still, because it is catabolizing a particular gene or a particular compound, or it is involved in a particular pathway, it is highly likely that the genes would be similar. So there will be variations, there can be variation, they can be identical also, but that there is no certainty that 100% it will be identical. There could be variation. So there can be a stable hybrids, there can be unstable hybrids, and these hybridization we can control by the conditions that we provide. So just remember that probe is something that what we need to identify, it should have some complementarity to it. So either it is a similar sequence or it is the same sequence from other species. A similar sequence if we do not have exactly same enzyme, but we know that biochemically this enzyme is a hydrolase, or biochemically this enzyme is a dehalogenase. So we look at the dehalogenases, we find out the consensus sequence, and then we use that consensus sequence to form the probe. Because it is a dehalogenase, then probably my gene of interest in my bacteria would also contain that contain that consensus sequence, and therefore I will use to probe, use that sequence to create a probe which is there. These probes are labeled because when these probes are going to hybridize to a particular sequence, they must give some kind of a signal. They must tell us that, okay, I am here, out of these clones I am bound to this particular clone. So they must carry a particular signal, and that signal, how we introduce into a probe is basically a labeling method, which I will talk about next. But this is what is showing. So what you do, you use a probe, non-specific binding is there, specific binding is there. So as you can see, then you do certain kinds of washes so that non-specific binding or very loose bindings are removed, but specific binding. So specific binding represents stable hybrids. Stable hybrids are formed when there is a higher complementarity between the two things, and therefore it is more likely that that is our gene of interest which is there. So you will do that, and then you are going to expose it onto the X-ray films or undergo any biochemical processes. So depending upon the signal method that this probe is carrying, you basically process that, or you analyze that signal. For example, in this particular picture, it is shown that it is exposing this membrane onto an X-ray film, and in the X-ray film you can see a black spot. So this black spot is basically a positive hybridization, because it did not hybridize to DNA present in the colony regions of any other clones, but only this particular clone gave a signal because the probe went and got bound to this particular colony, and this colony is showing a black signal. So this is the resulting autoradiograph. So this is termed as an autoradiograph which comes in. Now if we go back to our original plate and we look at the colonies, then when we can identify that out of these 100 clones or out of these 200 clones, these are the clones that are positive. That means these are the clones that carry our gene of interest, and therefore we only pick those clones and do the analysis part. So this is how basically the screening for the desired clone is done in this case. As I said, the important aspect to understand in this is probe labeling. So I hope you understand the meaning of the probe. The probe is a DNA sequence which is used to investigate or to identify or to find out our gene of interest. So this probe is generally labeled and carry certain signals, because after hybridization it is going to signal us that, okay, this is the recombinant clone which is there. Now when we talk about probe labeling, the labeling can be done with a radioactive marker. So radioactive marker is basically when a DNA molecule is usually labeled by incorporating nucleotides that carry a radioactive isotope of phosphorus, which is P32. So you understand that in a DNA molecule, the backbone of DNA molecule, the bases are joined together by phosphodiester bond. So a phosphodiester bond is shown here. So these phosphodiester, this is just this is a deoxyribonucleotide molecule that is shown here. So that DNTP is carrying 3 phosphates, and one of the phosphate is radioactive. So when it is going to combine in the DNA synthesis, in a strand, then this is going to emit radioactivity. This radioactivity we can, this is a signal. So we can identify that. For example, we can expose it to the X-ray film. This radiations will give X-ray film, and when we process the X-ray film in the presence of that silver nitrate solution, it is going to give a black spot. So we are going to identify it. So this is a radioactive marker. So we use DNTPs which carry phosphate, a radioactive phosphate. And when we introduce this into the DNA, then that DNA is going to be radioactive and give out a signal which is there. Now the methods involved in probe labeling. So we use radioactive DNTPs, but what are the different methods? So different methods, one of the methods is Nick translation. So what is Nick translation? So in normal DNA molecules also, these nicks can be present. So nicks are basically the gaps on one strand of the DNA where the phosphodiester bond is broken, which is termed as discontinuity sometimes, and also certain bases are missing. So this is termed as the gap, like it is shown in the figure. So you have got a double-stranded DNA molecule, but you have also got nicks in it. So what do? You fill these nicks, or you basically synthesize the missing part of the double-stranded DNA. So for synthesis, what do you require? A DNA polymerase 1. And what do you require? The nucleotides that are missing. So you add the nucleotides, which is deoxyribonucleotides. You add ATP, you add GTP, TTP, CTP, all the 4 nucleotides you add. But one of the nucleotide, for example, in this particular case it is showing, use a radioactive DNTP. So in this case, DATP is carrying P32. So all other 3 nucleotides would also be there. So what would DNA polymerase do? DNA polymerase is going to bridge this gap, is going to create sequence at these nicks, and it also causes strand displacement. So it fills this and it also displaces the original fragment which is there. So the DNA polymerase 1 is able to attach to the nicked DNA molecules which generally can occur in a cell and catalyze what is called as a strand displacement reaction. So they will not only fill this nick, but they will also replace the rest of the strand. And what do? They synthesize it by adding the complementary bases to one of the sequences, but in that way it is also replacing the DATP with the radioactive DATP. So now we will have a double-stranded DNA probe carrying phosphate group, radioactive phosphate at all the DATPs which are there. So it will become a radioactive probe. So this is one of the methods by which the labeled probes can be generated. Another method is what is called as end filling. So it is already, the name suggests, is filling the ends. So how do you fill the ends? For this, what you actually require, you use the probe which has been digested with restriction enzymes, so that on the 2 ends you have got cohesive ends. So when you have got cohesive ends, you are using, for example, in this case it is showing an EcoR1 sticky end. Then again, those overhangs which are there, you complete those overhangs. So you synthesize the complementary strand. For that, a clinofragment is used. What is a clinofragment? Clinofragment is a part of the DNA polymerase which does not have the nuclease activity. So there are subunits in DNA polymerase in which one of these strand causes the nuclease activity. So it synthesizes, but it has also the ability to cause the nuclease activity, so that for the proofreading mechanism which is there. Clinofragment do not have, because what happens is when it also has the exonuclease activity, then sometimes it can actually cleave the DNA also. So the probe DNA which we are using to create the labeled probe by the method of Nick translation can sometimes be cleaved, because the DNA polymerase also have the nuclease activity. It can cut the nucleic acids. So for that, in the end filling mechanism, we use a probe which is, for example, a digested fragment. We use the clinofragment which does not have nuclease activity, and we use again the DNTPs. So one of the DNTP is again a phosphate labeled, phosphorus, radioactive phosphorus labeled, phosphate labeled. So as you can see, you will again have a labeled probe. In this case, the ends would be labeled. Now you can again denature them. You can have single-stranded probe, and single-stranded probe you can add into the, for example, the solution containing the nitrocellulose filter. So that nitrocellulose filter, which is again carrying the single DNA molecule of the library that we want to screen, so that you can use. So this is the end filling mechanism. It can be used to label the DNA molecules that have sticky ends. Clinofragment fills in a sticky end by synthesizing the complementary strand. So here the word is written synthesizing the complementary strand and not the strand replacement technique. So DNA polymerase 1 can strand replace because it can synthesize as well as cause the nuclease to remove the nucleotides as well. The third method, which is very commonly used, is termed as the random priming. So what happens in case of random priming is that you have got a double stranded DNA probe, you melt that and you get a single stranded DNA probe, and then you add the hexameric oligonucleotides. They are just random oligonucleotides, and these oligonucleotides are going to bind random sequences onto the template probe which is there. So denatured DNA is mixed with a set of hexameric oligonucleotides of random sequence that will contain a few molecules that will by chance base pair with the probe and prime the new DNA synthesis. So they will randomly bound because they are random. Some of the molecules, if you have got a hexanucleotide, then 2 nucleotide, 3 nucleotide will be at least in the same manner as it is found in the DNA, and therefore it is going to bound to that molecule. There will be random binding of the RNA fragments, and after RNA binding you can again use the clinofragment as well as the nucleotides, with one of the nucleotide as radioactive nucleotide, deoxyribonucleotide, to cause the the new DNA synthesis, and therefore again you will have a double stranded probe. So this is a random priming method which is done, in which any double stranded DNA fragment can be converted into a probe which is carrying the DNA fragment as well as the signal which is there. So this is the random priming method which is there. This is what we talked about when we want to use the radioactivity, but there are very many limitations. We have to have a license, we have to have a permission from the committees that we can use radioactivity because they are hazardous. Then there has to be a lot of precautions that need to be taken, handling the radioactivity, disposing the radioactivity. So there's a lot of cautions which are required. So nowadays many non-radioactive labeling methods are also introduced. So I will quickly talk about the 2 labeling methods. One is the labeling with the biotinylated nucleotide. In this case, what happens is that you use a DNA fragment which you need to probe, and then you add what is a biotin deoxyuridine triphosphate which you are using. So you add these DUTP, so you are converting it into, when you are priming this molecule, then you prime these molecule by using DUTPs containing the biotin. Now biotin is a compound which has got a lot of specificity for another molecule which is called as avidin. So biotin, avidin, they are highly specific to each other. So on the DNA fragment which you want to use as a probe, you synthesize the new DNA molecule by using this biotin DUTP. Then biotin DUTP, you again make them melt single strand, hybridize it to the DNA library which we want to screen. As you can see, it will hybridize specifically to the desired clone, and then you can introduce the avidin molecules. Now these avidin molecules will be highly specific to the biotin molecules, and these avidin molecules are carrying a fluorescent protein, a fluorescence which is there. So when they are going to bind to the biotin, they are going to give rise to a fluorescence, and this fluorescence then can be measured. It can be detected. So it can be detected by any chemical method, or it can be detected in the fluorocytometers where basically the fluorescence can be detected, and therefore we can know that which is the recombinant clone present in the library. So this is the biotinylated nucleotide if you use for the synthesis of the other strand of the probe, hybridizing it to the DNA, and then avidin is specifically carrying the fluorescent protein and doing that. The last method in this is labeling with a horseradish peroxidase. Horseradish peroxidase is again an enzyme. So what you are going to do is you take the single strand DNA and you complex it with this enzyme which is a horseradish peroxidase. Now this probe is carrying this horseradish peroxidase or HP protein which is there. When you hybridize it to the library, it is going to bind complementary to the specific clone which is there. And when you add luminosol, then this peroxidase is going to reduce the luminosol and give rise to the light intensity. And again, this light intensity can be detected. So using the radioactive markers, which is basically the radioactive phosphate group, for example, or using different biochemical methods such as the biotinylated method or use of horseradish peroxidase, these probes can be labeled. These labeled probes in the single stranded form can be added to the DNA where it will identify our desired clones. So we today talked about the construction of the genomic library, the construction of the cDNA library, the complementary DNA library, and also we looked at the screening of the libraries, the colony as well as the plaque libraries by the method of hybridization probing using the radioactive or the non-radioactive markers. We will talk about the rest of the methods apart from hybridization probing in our next session. Thank you.